Hello everyone, my name is Nyun Namgung and I'm in charge of poultry R&D at the Age Holdings. First of all, I would like to thank Chungang University for giving me the opportunity to present and for preparing this seminar. The content of this presentation is summarized from my doctoral dissertation, and I would like to inform you that the study was conducted with the support of Bio and Pathway Intermediate. The topic I will be presenting today is the effect of dietary supplementation with lysophospholipid and sophorolipid on productive performance and nutrient utilization in poultry. Then I will start this presentation. This is the outline of the presentation. The introduction introduces the background and reason for conducting this study. Next, I will briefly introduce the two key lipid materials used in this study, lysophospholipid and sophorolipid. After that, I will present the finding from the two experiments, and finally, I will conclude with the summary of the key result. Let's begin with the introduction. This graph shows the total complete feed production in Korea and the volume of imported feed ingredients of the past 23 years. As you can see, complete feed production has steadily increased, and imported of feed ingredients have risen proportionally as well. Unfortunately, the domestic self-sufficiency rate for lower ingredients has shown a declining trend during the same period. Given that a large proportion of feed ingredients used in Korea are imported, the efficient utilization of feed resources become a top priority for the industry. Improve the efficiency of the feed ingredients, it is essential to enhance their overall utilization. Among the major strategies to achieve this, the use of exogenous enzymes such as NSPase, phytase, and protease is one of the most effective and widely applied approaches. Recently, functional complex lipids have been receiving increasing attention because they offer better cost effectiveness while providing effect similar to those of exogenous enzyme. Unlike enzyme that act directly on dietary substrate, lysophospholipid belonging to the phospholipid groups are known to enhance nutrient absorption by acting on the cell membrane of intestinal epithelial cells. Another complex lipid groups gaining attention is the glycolipid base sophorolipid. Recent studies have reported that sophorolipids positively affect various indicators related to the animal robustness. However, very few studies have investigated how these beneficial lipids influence actual productivities in poultry or livestock animals. Therefore, we became interested in evaluating the potential of lysophospholipid and sophorolipid as feed additives aimed at improving feed efficiency. And I would like to explain lysophospholipid. First, let me explain the formation process of lysophospholipid. Phospholipids are fundamental components of the cell membrane, consisting of phosphate-containing head groups and two fatty acid chain. When the enzyme phospholipase A2 acts on a phospholipid, one fatty acid chain, one fatty acid is hydrolyzed and removed, resulting in the formation of a phospholipid with only one fatty acid chain. This molecule is called a lysophospholipid. Lysophospholipids comes in many different forms. Depending on types of phospholipids they originated from. Among the major types, lysophosphatidylcholine choline is known for its effective on membrane permeability. Lysophosphatidylcholine can influence membrane fluidity, and lysophosphatidylethanolamine also affect membrane permeability. We also have lysophosphatidic acid, which helps stabilize membrane packing. Now, the exact composition of lysophospholipid products can vary depending on the quality of low phospholipid and manufacturing process. But if we take soybean lecithin as an example, LPC is usually the predominant component, while LPA tends to be present at the lowest level. Lysophospholipid promote nutrient absorption through several mechanisms. First, because they contain only one fatty acid, lysophospholipid can insert themselves into the cell membrane. As they enter the membrane, they change its physical properties. This insertion increases their spacing between adjacent phospholipid, which in turn enhance membrane fluidity and permeability. As a result, nutrients are able to cross the membrane more efficiently. At the same time, these structural changes stimulate the activity of membrane transport proteins, which further improves the absorption of molecules such as amino acid and glucose. Another well-known function of lysophospholipid is their emulsifying ability. Dietary fat must first be emulsified into small droplets by bile acid before they can be digested. When lysophospholipid are present, they support this emulsification process, allowing bile acids to work more efficiently. This creates a large surface area for lipase to act on, which improves fat digestion. Finally, the lipase digested products like free fatty acids and monoglycerides are observed across the intestinal epithelium. Next are the research results related to lysophospholipid. This study, published in Poultry Science in 2019, evaluated the effect of lysophospholipid in broilers. The experiment was conducted for 5 weeks using a total of 5 treatments with 6 replicates per treatment and 10 birds per replicate. The treatments included a positive control diet and a negative control diet formulated with 150 kilocalorie lower metabolizable energy than the positive control. In addition, 3 more treatments were created by supplementing the negative control diet with 3 different levels of lysophospholipid. According to the result, all lysophospholipid supplemented groups showed significantly improved feed efficiency and crude protein digestibility compared with the negative control. However, there were no differences among the lysophospholipid treatment based on the inclusion level. In the second experiment, presented in the same paper, the negative control diet was formulated by reducing crude protein and amino acid instead of energy. The result shows that feed efficiency and crude fat digestibility was significantly improved in the treatment supplemented with 0.15% lysophospholipid compared with the negative control. However, there were no differences among the different inclusion level of lysophospholipid. Overall, these findings indicate that supplementing lysophospholipid in poultry diet can improve the feed efficiency and nutrient digestibility, and can partially compensate for reduced dietary energy and crude protein level. Next, I will introduce sophorolipid. Sophorolipid consists of hydrophilic sophorous head, which is made up of two glucose molecules, and a hydrophobic fatty acid tail. Because of this structure, they are classified as glycolipid within the broader group of complex lipids. They are also known as natural biosurfactants, and these properties allow them to support fat digestion, similar to lysophospholipid. Recent researches has reported that sophorolipids can enhance cellular recovery and improve the intestinal bilious environment. They are also well known for their environment antimicrobial and immunomodulatory effects. Let me briefly explain how saprolipids are produced in yeast cells. First, inside a cell, when a fatty acid such as oleic acid binds subsequently with, with two glucose molecules, an acidic form of saprolipid is generated. This acidic form is then transported outside the cell, where it can be converted into the lactonic form through the action of lactone esterase. Both the acidic and lactonic form also exist in more detailed subtypes depending on whether they are acylated. In our study, we used a mixture of natural produced saprolipids from yeast, consisting of approximately 60% acidic form and 40% lactonic form. Saprolipids play a role in the body similar to other glycolipids. In animal cells, glycolipids usually make up about 5% of the total membrane lipids. Their lipid portion is embedded in the cell membrane, while the sugar portion is exposed to the outside of the cell. The function of glycolipids are largely determined by this exposed sugar region. Because this region acts as a recognition site between cells, glycolipids are involved in various types of cell-to-cell interaction. When external cells or molecules bind to this recognition site, they can trigger immune response or signaling process inside the body. Soprolipids are also glycolipid composed of a sugar head and a fatty acid tail, so they are expected to interact with the cell membranes in a similar way. Thus, soprolipids may help modulate immunities exert antimicrobial activity, and improve gut health. However, the exact mechanism remain unclear and require further research. Next, next are the research results related to sophorolipid. In this 2021 Journal of Animal Science study, broilers were fed either a control diet, 10 ppm vancomycin, or 10 ppm sophorolipid. Sophorolipid reduced the pro-inflammatory cytokine IL-6 and increased the anti-inflammatory cytokine IL-10. It is significantly upregulated CLDN1 and MUC2, indicating improved tight junction integrity and mucin production. Overall, 10 ppm of prolipid shows clear anti-inflammatory and gut health benefits. In the same study, the researcher also evaluated the antimicrobial activity of soprolipid. Supplementing broiler diet with 10 ppm soprolipid significantly increased the beneficial bacterial, such as Lactobacillus species and Akkermansia muciniphila, while effectively suppressing the harmful Streptococcus galactolyticus group. Taken together, these results shows that adding 10 ppm of soprolipids to broiler diet support immune modulation, strengthen the gut environment, and helps reduce pathogenic bacteria. Based on the effect of lysophospholipid and soprolipid we have seen, I expect is that combining these two complex lipids could result in a synergistic effect. To verify this, two experiments were conducted. The title of experiment 1 is the effect of dietary supplementation with lysophospholipid and superlipid on productive performance and nutrient utilization in broiler chickens under normal or heat stress conditions. A total of 2,560 one-day-old broiler chicks with no sex separation were used in this experiment. The treatment consisted of 8 dietary groups with 8 replicate each, and 40 birds were assigned per replicate. Here are the treatment group. Among the 8 treatments, 4 were raised under normal conditions, and the other 4 were raised under heat stress condition. Within each condition, lysophospholipid and sophorolipid supplementation was arranged in a 2 by 2 factorial design. For the measurement, we evaluated gross performance, small intestinal villus morphology, and the water holding capacity of breast meat. A cyclic heat stress program was applied for the heat stress condition. Throughout the entire experimental period, the temperature humidity index THI value in the heat stress condition remains in a danger zone, indicating that heat stress was successfully imposed. Here are the results. When you compare the normal condition with the heat stress condition, body weight gain, feed intakes, and feed efficiency were all significantly reduced under heat stress condition. These findings clearly shows that heat stress conditions negatively affected broiler performance. In both the normal and heat stress conditions, No significant differences were observed in productive performance. However, the mixture of lysophospholipid and sophorolipid shows numerically improved feed efficiency compared with the control groups, indicating a synergistic effect. Nonetheless, no differences were detected among treatment in body weight gain, feed intake, and nutrient utilization, and no interaction effects between the two factors were observed. In both conditions, villus height increased in the treatment groups compared with the control group treatment, and under the normal condition, this increase was statistically significant. However, no synergistic effect was observed in the combination treatment. For the water holding capacity of the breast meat, an interaction between environmental condition and supplementation factors were observed. Under the normal condition, water holding capacity decreased in the sophorolipid and mixture of lysophospholipid and sophorolipid treatment. However, under heat stress conditions, the combination treatment shows the highest water holding capacity, indicating that the combined supplementation was particularly effective on the heat stress, indicating a synergistic effect. Based on the results obtained so far, the conclusion of experiment 1 can be summarized as follows. Under both normal and heat stress conditions, supplementing lysophospholipid or sophorolipid alone did not improve the feed efficiency. However, supplementation with a mixture of lysophospholipid and sophorolipid led to a numerical improvement in feed efficiency. In the heat stress condition, the mixture treatment also enhanced the water holding capacity of the breast meat. In addition, lysophospholipid, soprolipid, or their combination increased villus height in the small intestine. In experiment 2, the effect of dietary supplementation with lysophospholipid and soprolipid on productive performance and nutrient utilization in laying hens were evaluated. A total of 384 Lohmann brown laying hens at 47 weeks of age were used, and the trial was conducted for 12 weeks. The treatment consisted of 4 dietary groups with 8 replicate each, and 2 rewards were assigned per replicate. The details of the dietary treatment are as follows. The positive control treatment was formulated to meet the recommended nutrient requirement for Lohmann brown laying hens. From the positive control diet, uh, negative control diet was created by reducing the metabolizable energy by 80 kilocalories per kilogram. 2 additional treatments were derived from the negative control diet. One supplemented with lysophospholipid alone, and another supplemented with a mixture of lysophospholipid and sophorolipid. In this experiment, the sophorolipid-only treatment was excluded because its synergistic effect could be assessed by comparing lysophospholipid alone with the lysophospholipid and sophorolipid combination. After the laying trials, a metabolic trial with 4 replicates, 4 treatment, and 8 replicates were conducted to evaluate nutrient utilization and the metabolizable energy value of the diet. These are the results for productive performance. There were no significant differences or trend among treatment in hen-day egg production. However, hens fed lysophospholipid or the mixture of lysophospholipid and sophorolipid showed higher egg production than the negative control. In terms of feed conversion ratio, shortly FCR, both treatments, lysophospholipid and the mixture of lysophospholipid and sophorolipid, tend to have lower FCL than the negative control. However, no synergistic effect were observed in the mixture treatment. Next are the egg quality results. Egg quality was measured using an electronic devices. Both albumin height and whole unit, key indicators of egg freshness, were significantly improved by lysophospholipid alone or with the mixture of lysophospholipid and sophorolipid compared with the positive control diet and negative control diet. Especially, albumin height was significantly higher in the combination treatment than in the lysophospholipid treatment, confirming a synergistic effect of the sophorolipid on lysophospholipid. In contrast, yolk color was significantly lower in the mixture of lysophospholipid and sophorolipid treatment, indicating that the use of lysophospholipid in combination with sophorolipid may negatively affect yolk color. Eggshell thickness, eggshell color, eggshell strength shows no significant differences among treatments. For nutrient utilization, ether extract digestibility tends to increase in both the lysophospholipid and the mixture of lysophospholipid and sophorolipid treatment compared with the negative control. Metabolizable energy was significantly lower in all treatments compared with the positive control. However, the lysophospholipid and sophorolipid mixture treatment showed higher metabolizable energy than lysophospholipid alone, indicating a synergistic effect. Calcium utilization tends to be lower in all treatments compared with the positive control. However, both the lysophospholipid and the mixture of lysophospholipid and sophorolipid treatment shows a numerically higher value than a negative control. Phosphorus utilization was significantly reduced in the lysophospholipid treatment. However, it was increased to the level of the positive control in the mixed treatment, indicating a synergistic effect. In summary, the result of experiment 2 can be concluded as follows. Supplementation of lysophospholipid alone over the mixture of lysophospholipid and sophorolipid in lower energy laying diets improved FCR and enhanced egg freshness quality. Furthermore, the lysophospholipid and sophorolipid mixture shows a synergistic effect by improving nutrient utilization and metabolizable energy compared with lysophospholipid alone. Therefore, co-supplementation of lysophospholipid and sophorolipid can provide compensatory benefits in lower energy diet. This is the final summary. Lysophospholipid, as a phospholipid, are known to improve nutrient utilization and enhance productivity in livestock animals. Sophorolipids, as glycolipid, support gut integrity and contribute to overall robustness. Because no studies have evaluated the combined use of these two complex lipids, we conducted this research and observed clear synergistic effects across various indicators in both broiler and layers. In broilers, co-supplementation of lysophospholipid and sophorolipid improved feed efficiency, enhanced intestinal bilirubin morphology, and increased breast meat water holding capacity under heat stress conditions. In layers, supplementation with lysophospholipid and sophorolipid mixture improved egg freshness parameters and enhanced fat and phosphorus utilization, while also providing compensatory effect in lower energy diet. Therefore, further research is needed to continuous uncovering and understanding the effect of these two functional lipids in livestock. This concludes my presentation. Thank you very much for your attention.